Genetics, the science of inheritance, is the study of how parent organisms pass on their traits to their offspring. And the principles that govern genetics determine the way that all life on Earth will be passed on to future generations. Genetic principles are complex, but their outcome, such as the fact that children resemble their parents, seem very simple and ordinary. Yet few of us realize that most of the food we eat, the flowers we enjoy, and even the existence of civilization itself are largely the product of centuries of attempts by human beings to influence the outcome of inheritance. Now let us look briefly at the historical background of the science of genetics. For thousands of years, ancient farmers unknowingly used basic genetic principles when they attempted to improve food plants. For example, a wild grass called maize occasionally produced plants with large seeds. These seeds were saved, and the plants that grew from them were bred with one another. By developing corn plants that could be used to provide a steady food supply, people were able to settle down, and towns developed. And so, in many different parts of the world, because of simple genetic manipulation of wild food plants, great civilizations were able to flourish. Eventually, civilization developed to the point that people expanded their genetic tinkering to include non-food plants, plants that were desirable simply because they looked or smelled good. And today, most of the large, elaborate flowers found adorning the world's gardens, like these hybrid roses, were developed many centuries ago from much simpler plants, like the wild roses seen here that were bred with other wild strains of roses that had flowers of different colors, or that had unusual arrangements of petals. Also, the people of long ago captured wild animals and bred them with one another, eventually producing domesticated animals with genetic characteristics that were superior to those possessed by the wild breeds. For example, they bred sheep that grew heavier woolen coats and produced more meat, chickens that laid larger eggs, and faster, more powerful horses. As we have seen, human beings have been taking advantage of genetic principles for thousands of years and have benefited greatly by doing so. And yet it was not until the 19th century that genetics began to be investigated in a truly scientific way. During the 1860s, this man, named Gregor Mendel, decided to carry out the first genetic experiments by growing pea plants in the garden of the monastery in Czechoslovakia, where he lived as a monk. Mendel wanted to learn what would happen when pea plants with different, easily distinguishable physical traits, such as pod shape and color, seed shape and color, and length of stem, were bred with one another. Mendel chose to work with pea plants because they grew to maturity rapidly. And although peas normally were self-pollinating, it was a simple matter to cross-pollinate or cross-fertilize them with other plants. To carry out cross-fertilization, Mendel collected pollen that carries the male reproductive cells from the stamens located in the flower of one plant and transferred it to the pistil, the female reproductive organ located in the flower of another plant. As a result, seeds were formed that contained inheritance factors from each parent plant. Mendel's first experiment was to see what would happen if pea plants with short stems were cross-pollinated with other short-stemmed plants. The results of this cross did not surprise him, for in the first generation, called the first filial generation by geneticists, only short offspring were produced. Next, Mendel decided to see what would happen if pollen from tall pea plants was used to pollinate other tall plants. This time, his results were not clear, because although the seeds from some tall plants produced only tall offspring, seeds from other tall plants gave rise to first-generation offspring that were a mixture of both tall and short plants. Mendel could not understand these results, so he performed another experiment. He planted just the seeds from tall plants that had produced only tall offspring. He used these pure-breeding tall plants as one set of parent plants and cross-fertilized them with the consistently pure-breeding short plants. The outcome of this experiment was just as surprising as the first, because no short plants at all grew from the seeds resulting from this cross. The shortness trait completely disappeared in the first filial generation of plants. In a further attempt to make some sense of these strange results, Mendel cross-pollinated these tall first-generation plants with one another, and their seeds were grown into second-generation plants. This time, Mendel found that exactly 1/4 of the new second-generation plants were short, and the other 3/4 were tall. And these results did not vary, no matter which of the pea plant's 6 physical traits he was looking at. To explain how cross-pollination affected plant height, Mendel postulated that each plant must contain 2 inheritance factors. Plants with 2 tall inheritance factors will be tall, or with 2 short inheritance factors will be short. Yet if tall and short factors are combined, the new plants will always be tall. In other words, Mendel believed that the tallness factor overpowered the shortness factor in these plants. Therefore, he stated that the tall inheritance factor is dominant and the short inheritance factor is recessive. A short time later, Mendel's inheritance factors were renamed genes, and genes were to become the focus of tremendous biological research in the years that followed. Mendel's work laid the foundation for the new science of genetics, and it was not long before geneticists began to search for the actual location of genes inside of cells. At first, their search was narrowed down to the cell's nucleus, and then, a short time later, chromosomes, such as those seen here, were proven to be the location of genes. Geneticists also discovered another interesting fact, namely that most organisms they studied possessed 2 complete sets of chromosomes, which they called the diploid condition of a cell. Because chromosomes are found in pairs, this means the genes they carry will also be paired. And so this discovery provided a simple explanation for why the inheritance factors described by Mendel always occurred in groups of 2 that were dominant or recessive. The dominant and recessive genes of parents are transmitted to their offspring as a result of fertilization, which is the fusion of the male and female sex cells. However, fertilization can only occur if the male and female sex cells are haploid, that is, if the sperm and eggs contain single, not doubled, chromosomes. For if haploid cells were not employed in fertilization, the number of chromosomes present in the offspring would increase with each new generation, and eventually their cells would become filled up with chromosomes, and all normal cell functions would cease. Fortunately, a unique class of cells, found only in the ovaries and the testes, called germ cells, can undergo a process called meiosis that takes care of this problem. Meiosis, also known as reduction division, accomplishes two basic things. First, it reduces the chromosome number of the germ cells by half by breaking up the diploid chromosome pairs. The second outcome of meiosis is that each chromosome of a diploid pair ends up in a separate haploid sex cell. And this fact is critical to how the sex of an offspring is finally determined. Now let us look at how sex determination takes place. Many organisms possess unique sex chromosomes that carry special genes which determine whether the offspring resulting from fertilization will be a male or a female. For example, out of a total of 23 chromosome pairs, human beings possess one pair of sex chromosomes. Human females have an XX pair of sex chromosomes, and human males have an XY pair instead. Now let us follow these chromosomes through meiosis. When a germ cell in the ovary of a female undergoes meiosis, 4 identical egg cells are produced, each with a single haploid X chromosome. But when a germ cell in the testis of a male undergoes meiosis, 4 sperm cells are produced. 2 will contain single X chromosomes, while the other 2 will contain single Y chromosomes. During fertilization, if the first sperm cell to enter the egg contains a Y chromosome, the offspring will be XY or male. But if the sperm cell has an X chromosome, the offspring will be XX or female. And so there is a 50-50 chance of being either one sex or the other. We have learned that chromosomes carry the genes which can determine not only the sex of an offspring, but its other physical traits as well. Now let us re-examine Mendel's work on pea plants by looking at what happens to their chromosomes and the genes they carry through 2 generations of crossbreeding. For the sake of simplicity, let us say that Mendel's pea plants possess only 2 pairs of chromosomes, an A pair and a B pair. The gene that regulates the plant's height is located on the A chromosome, whereas the B chromosome has no genes that affect height, but instead has genes that affect other physical traits, such as seed and pod color. True breeding tall plants, those that produce only tall offspring, will have 2 identical A chromosomes, each with a T gene for tallness. And likewise, the true breeding short plants will also have 2 identical A chromosomes, but each A chromosome in the short plants will contain the recessive gene small t for shortness. In this cross-fertilization, pollen from the true breeding tall plants will be used to fertilize the female sex cells of a short plant. Consequently, after meiosis, each pollen grain will contain a haploid male sex cell with an A chromosome that possesses a tallness gene T. And each haploid female sex cell will contain an A chromosome that possesses a recessive shortness gene small t. Following cross-fertilization, all of the new diploid seeds will be the same and will contain 1 A chromosome possessing a tallness gene and 1 A chromosome with a shortness gene. Since tallness is dominant over shortness, all of the first-generation plants that develop from these seeds will be tall. Now let us see what happens when the germ cells of these first-generation plants undergo meiosis. Following meiosis, half the male sex cells will contain tallness genes, and the other half will contain shortness genes. And the same will be true of the female sex cells. Now let us see what happens when we take each of these male sex cells and cross them with each type of female sex cell. The first cross will yield 4 tall plants, because the tallness gene is always dominant over the shortness gene, and a tallness gene is present in each of these offspring. The second cross, using the second of the 4 male sex cells, will also yield 4 tall plants. The third cross, using the third male sex cell, yields 2 tall plants and 2 short plants, because 2 of the new seeds contain the 2 recessive genes needed to create a short plant, whereas the shortness trait is masked in the 2 other offspring. The fourth cross, using the 4th and final male sex cell, produces the same results as the third cross, 2 tall and 2 short plants. If we tally these results, we will find that, out of a total of 16 plants resulting from these crosses, 12 or 3/4 are tall, and 4 or 1/4 are short, which is exactly what Mendel would have predicted. To simplify our understanding of this process, the format seen here, called a Punnett square, was developed early in the 20th century. Using this method, dominant genes, such as the genes for tallness, are given capital letters, and the recessive genes, such as the genes for shortness, are given small lowercase letters. The genes of one parent are found on the top of the box, and the genes of the other parent are on the left side of the box. To perform this cross, the upper left letter is multiplied by the first top letter. Then the upper left letter is multiplied by the second top letter, and so on. After completing this simple cross, we find that 1/4 of the offspring are capital T, 1/2 are capital T, small t, and 1/4 are small t, small t. This distribution of the gene for a pea plant's height is called its genotype, which is the actual True or false? Mendel found that when he cross-pollinated short pea plants with one another, the offspring were sometimes a mixture of tall and short plants. A gene such as the one for tallness in pea plants is said to be because it overpowers the recessive shortness gene. An organism's physical traits make up its phenotype, but its actual genetic makeup is called its If a human cell is found to contain an X and a Y chromosome, that cell must have come from a person whose sex is the biological process that converts diploid germ cells into haploid sex cells is called The science of genetics fully entered the molecular age in the mid-20th century with the discovery of the 3-dimensional structure of a giant molecule called DNA, or deoxyribonucleic acid. DNA is a major component of chromosomes, and by knowing its structure, it became possible for scientists to understand how genes store information inside of living cells. Furthermore, the discovery of the structure of DNA made it possible to understand how genetic changes can take place that can permanently alter the physical traits of an organism, as well as those of its offspring. Now let us look at chromosomes, genes, and DNA more closely. If we could remove a single cell from one of the pea plants that Mendel used in his experiments, and then examine in detail an A chromosome, where the gene controlling plant height is located, we would discover three basic facts. First, that the chromosome is constructed of large quantities of DNA. Second, that genes are composed of distinct regions of the giant DNA molecule, and so the gene controlling plant height is always found in exactly the same location on the chromosome. And third, we would discover that many other genes are also found in different but always predictable locations on this same chromosome. Today, scientists estimate that human beings, for example, possess around 100,000 genes located on 23 different chromosomes. So this would mean that the average human chromosome would contain about 4,300 different genes. Many decades of research have proven that virtually all genes have just one basic function. That is, to store the instructions for building proteins. It is now believed that there is a different gene for each different type of protein found within all living cells. Proteins are essential components of living things. Some help create structures, such as fibers and membranes. Others transport oxygen in the blood. But the vast majority of proteins function as enzymes. To be able to understand how genes control physical traits and how they can be dominant or recessive, it is very important to understand the role of enzymes inside of cells. To learn one typical way that enzymes work, we will use an imaginary biochemical system in pea plants as an example. Let us imagine that, in order for pea plants to grow tall, a certain chemical growth factor, K, must be present inside their cells. This growth factor is formed when two chemical subunits combine with one another. However, these subunits will combine only if a T enzyme is present to join them together. Otherwise, they will merely float around aimlessly inside the cell. The instructions for building the T enzyme are found in a small region of the DNA on each of the diploid A chromosomes. And as we have already learned, these identical DNA regions are the genes that control plant height. As we have seen, some tall plants have capital T tallness genes on each A chromosome, and therefore have the capital T genotype, while other tall plants have the capital T small t genotype, because one chromosome contains a recessive shortness gene. We can now see that the reason the tallness gene capital T is dominant is that it can direct the formation of the T enzyme that allows the two chemical subunits to combine to form the growth factor, even if the instructions are present on only one of its two A chromosomes. In contrast, if only the two recessive small t genes are present on each A chromosome, the cell does not possess the correct instructions to make the T enzyme. As a result, the subunits remain uncombined and no growth factor is created. And hence, in this imaginary example, the plant will remain short. Changes in genes like those that cause shortness in pea plants are called genetic mutations. Genetic mutations occur when a cell's DNA instructions are changed, either as a result of external events, such as exposure to chemicals or radiation, or by mistakes made in copying DNA during cell reproduction. To understand how genetic mutations occur, it is first necessary to know how instructions are stored by DNA molecules. To store information, DNA employs a genetic code forming words and sentences from chemicals to create a fantastic language of life that is employed by every living creature. The genetic code language is very similar to written languages that use an alphabet, but with these important differences. First, the genetic code alphabet is made up of only 4 different letters, A, C, T, and G, representing the 4 chemical subunits of DNA. Second, every word in the genetic code language is only 3 letters long. Third, with the exception of 3 punctuation words, all of the other genetic code words are the names of amino acids, which are the chemical components from which proteins are constructed. Fourth, a genetic code sentence of instructions is the same as a gene. And fifth, the order of genetic words in gene sentences determines the order of amino acids in proteins, and in turn, the specific order of amino acids in proteins is what allows them to perform specific tasks. To further understand these concepts, let us imagine that the gene for the growth factor enzyme, T, in pea plants consists of the following chain of genetic code words on the DNA: AAT, GGG, ATA, TTT, CGC. This is an instruction for 5 different amino acids to be placed in the correct order during the construction of this enzyme. For the T enzyme to function properly, it is absolutely essential that the correct amino acids appear in the correct order in the protein chain of the T enzyme. For if just one amino acid is incorrect, the protein structure will change, and usually the enzyme will no longer function properly, which in this case means that the growth factor subunits will remain uncombined and no growth factor will be produced. Hence, the plant will not grow tall. Incorrect enzymes like this are usually the result of genetic mutations that confuse the DNA instructions for making proteins. For example, the production of this incorrect T enzyme resulted from the change of just a single letter in the DNA gene sentence. In this case, a G was substituted for a T in the 3rd word of instructions, so that amino acid 7 was substituted for amino acid 3. As might be expected, this is called a substitution mutation. A substitution mutation can occur at the time the DNA is replicated, prior to cell division. This type of mutation is fairly common and may explain how, long ago, the pea's recessive gene for shortness was created. However, many other less common types of mutations also exist. For example, deletion mutations occur if parts of the gene are deleted during DNA replication. And insertion mutations can also occur during DNA replication if extra DNA is inserted into a gene. A final class of mutations occur when the chromosomes themselves are damaged or broken, or, as is the case with Down syndrome, an entire extra chromosome is present. If mutations such as we have just seen occur in the DNA of non-germ cells, the result can be cell death, tumors, or uncontrolled cell growth, such as occurs in cancer. These mutations, however, are not passed on to the offspring. But if a mutation occurs in the genes of germ cells in the ovaries or testes, the resulting sex cells will contain the defective gene, and following fertilization, that gene will be replicated over and over again as the cells of the fertilized egg divide. And eventually, every cell of the offspring will contain the defective gene. Then, if the offspring with the defective gene mates with another defective gene carrier, the gene will be present on each of the diploid chromosomes, and the most common results are death or genetic diseases. Sometimes genetic diseases are not very serious, as is the case with shortness in pea plants. But in other cases, they can be very damaging, as with the human genetic diseases, cystic fibrosis, and sickle cell anemia. We have seen how mutations can cause genetic diseases and death. But perhaps the most interesting fact about mutations is that they also have a positive side, because sometimes they are helpful and have provided living organisms with an extremely successful method for producing biological changes. Let us see how this happened. Most biologists believe that life first appeared on Earth about 3.5 billion years ago in the form of organisms similar to the bacteria seen here. Over millions of centuries, these primitive organisms were subjected to powerful forces from a continually changing environment. Mutation after mutation occurred, and as a result, organisms took on different physical traits. Organisms possessing the most favorable mutations, those that allowed them to live under particularly harsh conditions, survived to produce offspring, and those that lacked these favorable mutations died out. Eventually, organisms grew more complex as favorable mutations caused even greater physical changes to occur as creatures adapted to life in every imaginable environment. Over time, the variety of living things continually increased, and today the number of different species recognized by biologists is in excess of 1,400,000. And yet all of these creatures, from single-celled protozoans like this, to complex multi-celled creatures like this butterfly, employ exactly the same genetic code language to store the information upon which their lives depend. And today, scientists are actually able to produce genetic mutations in living creatures, and thereby create entire new varieties of organisms that possess unique combinations of traits never before seen on Earth. The English alphabet has 26 letters, but the genetic code alphabet uses only letters. Genes are distinct regions of a very large information storage molecule, known by the abbreviation Nearly all of the three-letter genetic code words used to write gene sentences are actually the names of the protein components called most chemical reactions inside of cells, such as the joining of the growth factor subunits, will only occur if the type of proteins called are present. Errors in genes that result in the production of defective proteins are called